Hello everyone, welcome to my channel. So today we are going to discuss about dark reaction of photosynthesis. We know that photosynthesis is not just a one-line reaction, rather it is divided into two reactions, light reaction and dark reaction. In the last class we have already discussed about the light reaction. So what are the products of the light reaction? We can see that is going to be ATP. Along with that we have also seen NADPH as well, and in the light reaction, we see that oxygen is also evolved. Now, oxygen is not going to participate in the dark reaction, but the products of the light reaction, such as the ATP and NADPH, they are going to participate in the dark reaction, and we can see Calvin cycle, that is C3 cycle, is going to occur. So we can see this process, that is dark reaction, it is independent of light, but it is dependent on the products of the light reaction such as ATP and NADPH. And where it is forming? It is forming in the stroma region. So this fluid filled structure that you can see, I have also marked it here. This is what we call as the stroma region. Okay. Now, during this dark reaction, we can see it is also called as biosynthetic phase and Blackman's reaction. So we will see that the carbon dioxide, it is going to be converted into glucose, that is 6 carbon molecule compound. So we are going to fix this carbon dioxide. For fixing the carbon dioxide, we use an enzyme that is called as Rubisco, that is ribulose 1,5-bisphosphate carboxylase and oxygenase. So you must be wondering, this enzyme can also do carboxylation and oxygenation? Yes, it can. But it depends that which activity it is going to do, because it is going to have both carboxylase and oxygenous activity. So that means that the carbon dioxide and the oxygen, they are going to compete for the same active site of the enzyme. And just to give you an idea that this enzyme is the most abundant enzyme in the world. So how does it happen? When do we see Calvin cycle occurs? Whenever there is high amount of carbon dioxide in the air, that means you can say that at low temperature and also the high carbon dioxide is to oxygen ratio will be there, then only we see that Rubisco behaves as carboxylase. On the other hand, if we see that if the enzyme acts as an oxygenase, that time we will see there is going to be high temperature and low carbon dioxide and oxygen ratio, and that is the time when we see the C2 cycle or photorespiration occurs. Okay. Why photorespiration? Because it is happening in the presence of light, and we will see the products are similar to our respiration. We will be discussing about it. But before that, let us now focus on Calvin cycle, that we also call as the C3 cycle. So you can see there are three stages. First, there is going to be carboxylation using the Rubisco 1,5-bisphosphate carboxylase oxygenase, but it is going to work as carboxylase here. And we are going to use the 5 carbon. So I will be writing the carbon number of each of these like compounds which are going to be utilized. So 5 carbon ribulose 1,5-bisphosphate, and we can see that the oxygen, that is the carbon dioxide, is basically used here. So it is 1 carbon. So we can see 5 carbon plus 1 carbon, it gives 6 carbon, right? But 3-phosphoglycerate, that is 3-PGA, is not 6 carbon, it is 3 carbon. That means 2 molecules are going to form. We can continue this reaction like this. But students, I remember that for the starch formation or for the glucose formation, there is going to be 6 carbon required, right? Because glucose is C6H12O6. We know about it. That means how many carbon dioxide will be fixed? 6 carbon dioxide will be fixed. Okay. So why don't we start the reaction with the 6 carbon dioxide? So it is going to be easier for us. Yes, we know that hard work is important, class, but why don't we switch to smart work, right? So instead of taking only 1 carbon dioxide, from now onwards, I am going to take 6 carbon dioxide and 6 molecules of ribulose 1,5-bisphosphate. So what we can see, ribulose 1,5 RUBP is now 30 carbon plus the 6 carbon from the carbon dioxide, right? It is going to be 36 carbon, right? I hope it is clear to you all now. So that means here the 3 carbon compound that is going to occur, we have to just divide, right? 36 divided by 3, 12. So that means 12 molecules of 3-PGA. Okay, so we will see. Let me write it here for all of you. 12, I am writing in small molecules of 3-PGA. 3-PGA basically means 3-phosphoglycerate. Now we can see it is going to form 1,3-bisphosphoglycerate. Okay, and here there will be ATP used. So of course, if it is 12 molecules are there, so obviously we will see 12 ATP is going to be used. Now reduction will lead to formation of triose phosphate. Triose phosphate is a 3-carbon compound, but the triose phosphate will help in formation of 6 molecules, that is 6 molecules, not rather we say 6 carbon compound, that is glucose will be formed. That means from this 36 molecule we have to subtract 6 molecules, and that is giving us 30 carbon. So as the 2 molecules of triose phosphate is utilized, so after this, like reduction, in this reduction process we will see triose phosphate is forming. It requires reduction and it requires 12 NADH. Okay, so this is NADPH, and NADPH is going to be utilized 12, and it is forming triose phosphate. Now the triose phosphate is 3 carbon compound. So what? How many molecules will be there of triose phosphate? Very easy. 30 carbon is there now because 6 are being utilized here. Now 10 molecules of the triose phosphate. The triose phosphate forms the ribulose 5-phosphate, which is a 5 carbon compound. So it is forming 6 molecules of ribulose 5-phosphate. Okay, because we have to fix it that 30, right? 6 into 5 for 30, that is 6 molecules of ribulose 5-phosphate. Now this ribulose 5-phosphate is going to form into ribulose 1,5-bisphosphate, that is ATP will be required. So how many ATP? We will see 6 ATP will be required, and there is going to be regeneration of the ribulose 1,5-bisphosphate. Okay, so we can see that how in Calvin cycle carboxylation is done by Rubisco. Then we can see reduction, it is forming triose phosphate, and then triose phosphate is giving the 6 carbon compound, that is glucose, utilizing how many carbon dioxide? 6 carbon dioxide. So we can say that the Calvin cycle is moving 6 times. So 6 times Calvin cycle. What are the things that are required? So if we see the ATP we can see that there is going to be 6 plus 12. So that means 12, 8, 6 plus 6 plus 12, 18 ATP, and we have the NADPH. How many NADPH are required? We can notice here that NADPH is just 12. So this is the 6 times Calvin cycle, right, students? So let's say if we want to do it for one turn of the Calvin cycle, what can be done? So we can see 18 ATP is utilized. So for one turn, we are going to divide 18 by 6, and that is coming as 3 ATP. And NADPH, okay, so let me write it is not NADH. It is going to be NADPH. So how many NADPH is required? That is 12 divided by 6. It is going to be around 12 NADPH. I hope it is clear in one turn of Calvin cycle how many NADP and also ADPH and also one ATP, and how many ATP in one turn of Calvin cycle, that is 3 ATP and 2 NADPH. Okay. Now students, if you all remember, we also know about the oxygenous activity. So when there is oxygenous activity of Rubisco, we see there is C2 cycle. We also tell it as C2 cycle because last time we saw that the product that was forming, it is a 3 carbon compound, that is why C3 cycle. Now the product that is going to form, it is 2 carbon compound. Okay, so that is why we are going to call it as the C2 cycle. So C2 cycle is forming when the Rubisco is performing oxygenase activity, and here we will see loss of 2 carbon. Why so? Because RUBP, which is a 5 carbon compound, it is going to form a 3 carbon compound, right, and 2 carbon carbon compound. But why I am telling the two carbon compound, that is 2-phosphoglycolate, it is not going to be useful, or we can say there is a loss, because it cannot participate in the Calvin cycle. Rather, 3-PGA, it can participate. So what will happen to this 2-PGA? Will it not work? No. Let's say if we are seeing that there is 2-RUBP that is going to participate, okay, then what we will see, that there is going to be how many molecules? So we will see there is going to be 2 molecules of 3-phosphoglycerate, and we can also see there is going to be 2 molecules of, so we will see there is going to be, okay, I hope all of you cannot see this. Okay, just a second, let me see what can be done. So we are going to see that there is going to be formation of again 2 molecules of 2-phosphoglycolate. Phosphoglycolate. So if there is 2 RUBP, we know that there is 10 carbon now utilized, right? So out of this 10 carbon, we are going to see that 6 carbon is going to be participating in the Calvin cycle. But what about the 4 carbon? Because we know it is a 2 carbon compound, so total 4 compounds, we are multiplying by 2. So 4 carbon, will it be lost? No. Out of this 4 carbon, students, we can get back 3-PGA as well. So you must be wondering, ma'am, how we can get back this, like from the 4 carbon we can get? For this, we have to understand the photodesperation process. Yes, it is like wasteful product, but if we see that there is going to be 2 RUBP that is going to be utilized, so that time we can see that the product won't be fully wasted, but yes, it is still going to be wasted. So RUBP along with the oxygen using the Rubisco forms 3-PGA, and we can see it can form 2-3-PGA if there are 2 RUBP utilized, and we can see there is phosphoglycolate, so there will be 2 molecules of this. So out of these 2 molecules, so that means 4 carbon. So we have noticed when this cycle is continuing 2 times, so glycolate is forming glycine, and when 2 glycine are formed, we can see there is going to be release of a carbon dioxide. So out of this, if one is released as a carbon dioxide, so we will be left with 3 carbon, right? So we will see this forming serine. After serine, it is going to enter the peroxisome. In the peroxisome, glycerate is forming, and glycerate is going to form 3-PGA. So we can see 3-PGA is going to form, right? So what we can see, students, out of the 10 carbon of the RUBP, we can see here that 3 carbon compound is got by the, we are getting that is 3-PGA, and 6 carbon which has been already, we have seen, we are also getting it back, right? So this is also being fixed, 3-PGA, 2 molecules of 3-PGA. But what is lost? Only 1 carbon is lost, and that is lost in terms of, that is our carbon dioxide. 1 carbon is lost. I hope it is clear to all of you how there is a 10% loss, right? Because only 1 carbon out of the 10 carbon we can see it is being lost. So we can say, so we can say there is 10% loss in the photorespiration process, okay? So that is why it's a wasteful process. But plants, like some plants, let's say they don't want this photorespiration to occur. So these are the C4 plants. They do not want this to happen. So in this plant way, we see plants, they are going to concentrate the carbon dioxide near the rubisco. So if the carbon dioxide is concentrated near Rubisco, so they are going to suppress the oxidation process, of course. And such like seafood plants include the maize, sorghum, sugarcane, and some plants of the tropical and the subtropical like region. But you must be wondering why is it happening? Because they have a anatomy which is called as the Kranz anatomy, and also they have dimorphic chloroplast, and they can also tolerate high temperature and show response to high light intensity. So that's really great because they are living in a tropical and subtropical climate. So what is the significance of this transdermal anatomy? We can see greater productivity of biomass because there is no loss of carbon, right? And reduced photorespiration. Yes, that will be great. And why so? Because carbon dioxide is constantly pumped into the bundle sheath of the mesophyll. So we can see there is a mesophyll cell and there is a bundle sheath cell which is involved here. So how it is so different from a C3 leaf, right? So let's say we have taken a C3 leaf. Here we can see the mesophyll cells. They are having the chloroplast. It is present in the palisade parenchyma. You all can see here, and it is also present in a spongy mesophyll. Okay. But in the C4 plant, there is no such like division. Okay, it is not differentiated. Mesophyll cells tissues are undifferentiated. So spongy mesophyll that you can see, it is present, but we can see there is a modification. Bundle sheath cells are also having the chloroplast, and palisade tissues are absent totally. Okay. So what is the difference between a mesophyll cell and the bundle sheath cell that is present in a C4 cycle? So we can see the mesophyll cell, they are thin and pervious to gas, so gases can enter easily. And it is also having less chloroplast and randomly arranged. It is having PEP carboxylase, which is very important for primary carboxylation of the we will notice. Okay, primary carboxylation is important. We will be seeing now. So there is also chloroplast which is having grana. So we know that granal chloroplast, it is having grana thylakoid and also stoma thylakoid, that is the lamellas. So both PS1 and PS2 is present here. Now there is going to be no C3 cycle in the mesophyll cell. It is made in a such a way. And in the bundle sheath cell, if you see, it is thick wall, impervious to gas. That means oxygen cannot enter as well. It is impervious to gas, and more chloroplast is present, centrifugally arranged as well, and it is lacking PEP carboxylase. So there is no carboxylation due to PEP carboxylase. But what it is having? It is having Rubisco. Okay. In bundle sheath cell, we will see Rubisco is present, which is not presented. No Rubisco in mesophyll. This is also a point we can add. And we can see chloroplast is without grana. So if it is a agranal chloroplast which is present in our bundle sheath cell, we will see that there is going to be only PS1. And students, what do we know? In PS1, is there any oxygen evolving complex? No. So as there is no oxygen evolving complex, we are going to see that there is going to be no oxygen evolved as well in the bundle sheath cell. So there is no chance we are concentrating or putting any kind of oxygen inside the bundle sheath cell so that we can concentrate only and only carbon dioxide. Okay, and C3 cycle occurs here. So how does it happen? So this is our mesophyll cell. Okay, this is our mesophyll cell. And this is our bundle sheath cell. All right. So we will notice that the mesophyll cell here, the carbon dioxide is entering. We have phosphoenol pyruvate. So the phosphoenol pyruvate along with the enzyme, that is PEP case it is written, but the full form is PEP, that is phosphoenol pyruvate carboxylase. It is going to do the fixation of the carbon dioxide, that is the first carboxylation, or we can say primary carboxylation, which is taking place in the mesophyll cell, and it is going to form OOA. And what is OOA? It is oxaloacetic acid. Okay, you all can write oxaloacetic acid. So this is the first product that is formed in the C4 cycle, and it is a 4-carbon compound. So that's why the name is C4 cycle. And after that, it is going to form the malic acid. The malic acid is going to be transported to the bundle sheath cell. And how it is transported? With the help of plasmodesmata. So students, you can see that through some channels it is coming. So these channels are called plasmodesmata, which are present in the plant cell. Okay, so now as it entering into the bundle sheet cell, we can see that malic acid has entered. Now there is going to be again decarboxylation. In decarboxylation, carbon dioxide is released. So you can see how we are concentrating from this C4 acid. Okay, this is a 4 carbon acid. We are going to do decarboxylation. We are going to get a that is carbon dioxide, which is going to participate in the Calvin cycle, because Rubensco is only present in the bundle sheet cell, not in the mesophyll cell. Okay, and we can see there is a 3 carbon compound that is forming, that is pyruvic acid, which is again transported into the mesophyll cell, and we will see that pyruvic acid along with 2 ATP is going to form our phosphoenol pyruvate. So we can see in C4 cycle, students, C3 cycle occurs. Along with that, we can see there is going to be also transport as well, right? We can see how the carbon concentrating mechanism taking place. So students, in the C4 cycle, if we see C3 cycle also takes place, that means how many ATP is being utilized? Firstly, we know in C3 cycle there is going to be how many ATP. So if we see here, for 1 carbon dioxide, it is requiring 3 ATP. So of course we have to write it is going to require 3 ATP, plus we also have to add this 2 ATP, which is required for the formation of phosphoenol pyruvate. So 2 ATP here. So total we can see 5 ATP is required. Okay, and the NADH we can see it is the one we can see the same here in the formation of the C3 cycle as well. So, but how many ATP? We can see it is requiring 5 ATP. All right. So now that we are clear up till this, now we are going to discuss about the CAM cycle. So we have discussed that how the carbon dioxide is concentrated. These are the certain plants in tropical and subtropical climate. But what about the xerophytes? Okay, how does C3 cycle also takes place here? So one thing, if you all notice, in C3 plants, C3 cycle occurs. In C4 plants also C3 cycle, Calvin cycle occurs. And also in CAM also it will occur. But as they are xerophyte, so what happens? There is reduced, we can say there is less rainfall there in those climate, in the desert climate. So that is why they are going to stop the excessive loss of water by transpiration, and for this they are going to close their stomata during the daytime. Okay, and that is why the gases, if they have to enter into the stomata, it can only enter during the night time, because at night time their stomata is open. So what we will see that here it is occurring in Opuntia, agape, like these are various xerophytes, and the name is Camplan because it is mostly common in Crassulacean family. Okay, so that is why it is named as Crassulacean acid metabolism. Now we will see dark acidification. How it is taking place? Because we will see there is going to be formation of malic acid. So during the daytime, we will see that the starch is going to be produced. It is going to break down and form triose 3-phosphate. It is going to form phosphoenol pyruvate, and phosphoenol pyruvate along with the carbon dioxide which is entering during the night time, it is going to form oxaloacetic acid. It is going to form malic acid. The malic acid is going to be stored in the vacuole during the night time. And students, the starch is present in the chloroplast. This is the chloroplast, just to give you an idea. Now, the malic acid is now present inside the vacuole. Now, the daytime. You can see the stomata is closed. Okay. So, we will see from the vacuole, the malic acid will come out, and it is going to form, like we will see the malic acid is going to break up and decarboxylation takes place. So, carbon dioxide is released and pyruvic acid. Pyruvic acid is the 3 carbon compound we know. So, the carbon dioxide, can it exit through the stomata? No. It cannot, and as it cannot, so carbon dioxide will be participating in the Calvin cycle. We have been studying that carbon dioxide is going to form the starch, that is the glucose, right, in the Calvin cycle. So through the Calvin cycle, it is also going to form starch, and also the pyruvic acid will be forming phosphoenol pyruvate. It is also going to form the starch. So that is how we can see the CAM cycle is helping for helping the xerophytics or the succulent plants to also perform the C3 cycle as well and form the starch as well. Okay, so these are certain ecological adaptation they also have. So I hope all of you are clear about C3 cycle, C4 cycle and the CAM cycle. And one thing is very common, that is C3 cycle is present in all of this with little bit of modification. Okay. So thank you for attending today's class, students. Have a nice day, everyone. Take care. Bye-bye.